Tonight's focus is going to be an introduction to genetics. Genetics is playing a larger and larger role in cancer research and, in fact, prevention and treatment of cancer. And what we're going to learn this evening really is just going to scratch the surface of that. But it is by no means a small part of the hereditary cancer community's makeup, and it's definitely growing in importance. If anybody has the opportunity to attend conferences such as the American Society of Clinical Oncology Conference, you will hear BRCA as well as genetics mentioned in almost every session these days. So our speaker this evening is Dr. Ora Karp Gordon. She is with the Cedars-Sinai Medical Center and has a long list of amazing titles, including Director of GenRisk, the Adult Genetics Program. She's the co-director of the Gilda-Radner Hereditary Cancer Detection Program and the Genetics Director at the Wassman Breast Cancer Risk Reduction Program. You'll see some additional accolades there. Her focus really is on hereditary cancers and the prevention of breast cancer and genetic disorders. And she has been involved in numerous clinical trials for cancer prevention. We're very, very fortunate to have Dr. Gordon with us here this evening. So I'm going to pass this over to her. As with our prior webinars, if you have questions, please feel free to enter them in the question or chat box. We will keep those questions and hold them for the end. And if for some reason the webinar runs a bit long, we'll address as many as we can, and then we will submit those questions to Dr. Gordon so that they can be answered and posted on the FRAT webpage at a future time.
Okay. Good evening, everybody. What I'm going to do over the next hour is just give you an overview and hopefully some language skills really related to genetics, the understanding of how genetic mutations affect disease, talk a bit about BRCA-related risks in cancers, as this will help you as you go out as advocates, whether you be on IRB committees, whether you're reviewing research proposals, whether you're out in the community. So an important thing to always hold, and I find that is very amazing, no matter how many times you ask the question, there are really a great deal of perceptions related to hereditary cancers, about genetic testing, ranging from, tell me everything, I want to be tested for everything, people walking with their arms stuck out, to tremendous fear and concern about violation of privacy, about, this sort of encroachment on things that are preordained. So I think you always need to sort of keep that in your mind when you're talking to both patients, to looking at potential research and how that affects society and the community at large. So the human genome, when I was in medical school, we were taught that there were 100,000 genes. Now there's probably less than 26,000 genes, and every 6 months or so I have to, change this slide to downgrade, the number. Is it that we're losing genes? Certainly not. What we understand now is that there actually are much fewer genes than was originally thought, and that number came from the number of proteins. So we could identify about 100,000 proteins. Turns out that there are much fewer genes that create, through sort of the Cuisinart of manipulating the DNA into a whole bunch of different products and functioning in different tissues. What we describe DNA as a sequence, so it's just an order of specific nucleotides, which are protein products, and all of our DNA is exceptionally similar. 99.9% of our DNA is the same from one person to another, regardless of what race or creed or where we come from in the world, or what our health or disease risks are. So this very small variation accounts for an enormous amount of variety in human trait and disease. And we'll talk about mutations, meaning changes in the DNA that, in fact, create an inability for a normal protein or a normal function within a cell and that this is what we understand to be hereditary, both in hereditary cancers and all sorts of other hereditary diseases. And then lightly at the end, I will touch on something called variation or polymorphisms. And that's important to sort of understand the distinction in the language, because you'll see a lot of that as well, both in the lay literature as well as any research or a conference endeavor you might attend. We all carry deleterious mutations. People also often feel very stigmatized when they find out that they have hereditary cancer. If they're carrying something they could have passed on to their children. But we all carry at least four or five deleterious mutations, some of which make us vulnerable to disease. Some of us would only make vulnerability to offspring should we marry somebody. We'll talk about that as we go on as well. Oh, excuse me. Okay, so yeah, dig back in your mind and sort of the segregation of traits, and if you sort of remember whenever that science class was with the Punnett square or the segregation of blue eyes and brown eyes, the principle is the same. And so we call these alleles or these traits, and they can segregate just related to an attribute or a disease risk. And it can be something that's dominant or recessive. If it's dominant, it means that you only have to carry one change in order to have either the manifestation of that disease or risk and pass it on. If it's recessive, there have to be two copies of this abnormal functioning trait or allele. The genetic code is indeed just that. It's sort of a series of codes, the details of which I think go beyond the scope of what most people need to do in their work as advocacy. But I think it just sort of helps to have a visualization of what's happening. So you see a very long strand of DNA, and it's made up of these repeating nucleotides or base pairs, these letters that you see, A, U, C, T. And they're read in sets of 3. And each of those then in turn tells an amino acid, which is the building block of a protein, okay, you have to get in line next. And so it goes, and there's a point of starting and a point of stopping. So a gene is just that series of coding that says these amino acids in this order can then become this protein, this enzyme, this transcription, this regulator of cell processing. Here's a schemata from the ASCO set, which is the Society of Clinical Oncology. And so you can see, so the DNA, our double helix, it unwinds. This one side is then read, and these amino acid chains then are demonstrated here as a protein, which is this sort of beaded little curly Q. And every protein has its own shape and three-dimensional configuration, and it has to be exactly right or it doesn't work properly. So if there's a mutation, noted by this sort of splat mark or star, it might change. It may foreshorten that protein. It may keep it from bending into the exact shape that it needs to be. And depending on how abnormal it is, there may be no protein, there may be reduced protein, or protein that just doesn't work as effectively. When we talk about the genetic basis of disease, there's sort of the macro view, which was the earliest ability for us to understand genetic disease, and that was actually in chromosomal disorders. So chromosomes are units of our genetic material, and we have 23 pairs of those. We get half from our mother and half from our father, and they're aligned sort of by the biggest size to the smallest size. They also have the gender X and Y. Females have 2 X's, males have an X and a Y. So disorders like Down syndrome, a form of mental retardation called Fragile X, and a variety of other chromosome abnormalities can be seen under the microscope because there are extra or missing chromosome material. If you ever had an amniocentesis because you were older than the age of 35, or you've had a screening test during pregnancy that was abnormal, it is generally looking for these chromosomal abnormalities. Single gene disorders like hereditary breast ovary cancer syndrome is due to a single mutation. You cannot see that looking under a microscope looking at cells specifically. You have to sequence the DNA to find this genetic change. So listed here are the HBOC, Marfan syndrome, which was the Abraham Lincoln kind of disease that is also a dominant disorder. So if you carry that change, you manifest it, and then you have a 50% chance of passing that on. as well as some common recessive diseases, which have no physical characteristics or health risks for that individual. Genomics, which is sort of the new generation, and where a great deal of research effort is being attended to now, is really channeling towards complex diseases, trying to understand what has been designated as sporadic cancer, the common breast, colon, prostate. What is that genetic vulnerability when somebody doesn't have a single gene mutation that we can find, as well as all other common diseases of aging. Another application of the genomics is related to pharmacogenomics or pharmacogenetics, as well as the prediction for cancer recurrence and treatment. And I'll talk about all these a little bit as we move on. So when you try to get used to the language of a mutation, we describe them related to how much they impact how that sequence is read. So the first line, you see the big red dog ran out. Then the nonsense, or the stop codon, the stop mutation, it says the big red, and then it stops. Frame shift, these deletions or insertions. So if you put in the wrong letter, suddenly the whole reading frame changes, and then everything is just gibberish after that. So you can see that the nonsense, the deletions, and insertions would clearly have the biggest impact on how that protein configuration would be affected. The missense mutations are more challenging to interpret. The big rad dog ran out may not make sense to a grandmother, but certainly might make sense to a teenager. So depending on the cell and how that functions, it may or may not have an impact. And this is one of the challenges in interpretation of genetic testing, because just looking at a change in the sequence doesn't automatically tell you whether there's a functional consequence. This also goes for these things called SNPs, or single nucleotide polymorphisms. So that's where there's one single base variation from one strand to another that is relatively common. The more common these changes are, the less likely that they're actually going to be disease-causing, but they could have some minor variation in functionality. This has been the source of a great deal of what are called association studies over the last 10 years. And often when you read, in the Times or whatever, that there's an association between this polymorphism and risk for alcoholism, obesity, cancer. These are the kinds of variations that they're talking about, that they're looking at one population versus another. Genetic heterogeneity is a concept that's just important to keep in mind, because there is not just one cause for one disease. Sometimes there is, but much more often there can be mutations in entirely separate genes that may be on completely different chromosomes and manifest in the same way. We know this to be true for hereditary breast ovary syndrome. BRCA1 and BRCA2 are on distinct chromosomes, but their function is very similar, and therefore the tissues that they're expressed in are similar. There are certainly differences in the cancer risks with BRCA1 and BRCA2, and that has to do with sort of nuances between the two genes and their protein effects, and the genes that the other genes that they interact with specifically. So you may have heard, and you certainly will hear, that all cancer is genetic. And that can be very confusing, because people think then that all cancer is hereditary. But there is a difference between the genetics of a cancer, the tumor itself, and the genetics of cancer susceptibility. So what is cancer? Cancer is unregulated cell growth. So cells that are supposed to stop dividing continue to divide. They no longer conform to their normal shape. They encroach on their neighbors. They overgrow. They can break free and grow someplace else. And that's what metastasis is. So cells growing on top of each other form tumors, and cells that break free and grow in places where they're not supposed to be growing, like from the breast to the bone, cause metastasis. What causes that is a series of mutations. So this slide demonstrates sort of the difference between what we call germline, or hereditary mutations, and somatic, mutations that are occurring just in the cell that ultimately is the progenitor, or the original cell that leads to the cancer. The germline, if you look to the left, is present in the parent, in the egg or the sperm, and in that child, all of the cells in that child and that baby have this mutation. That doesn't mean every cell in that body is predisposed to cancer, and it doesn't mean that every cell in the body is going to turn to cancer by any means. But it means that that individual is one step, one sort of giant step in a progression of injury that has to happen to any cell in order to have the potential to form cancer. In a somatic mutation, that hit or that first mutation occurs by chance due to carcinogenic exposure, to radiation, to aging of the cell, just issues over time. And once that first change happens, the cell has to acquire second, third, sometimes up to 15 or more injuries before it completely loses its self-regulation. The somatic mutations have a lot of focus now in terms of what we call targeted therapeutics or companion diagnostics. You look for a specific mutation in a tumor that's not been inherited from the parent and isn't passed to other family members, but that cancer itself may make it very vulnerable or responsive to a certain chemotherapy, or can be very predictive that this is a particularly aggressive type of cancer. So in this way, you can see sort of this progression, and we call this the Knudsen two-hit hypothesis, that you start off in the upper segment of the slide, you have two normal genes, and then over time, so these are the alleles, you get the two copies, one from the mom and one from the dad, and then there's an injury and one becomes no longer working. The cell works fine with just one copy. If by chance there are two damaged copies, then that cell starts to lose its regulation and can progress to a tumor. In hereditary cancer, you see that you're sort of one step ahead, and that just makes that much more likely that the second event will happen. Excuse me. So HBOC and most cancer susceptibility syndromes are what we call autosomal dominant with incomplete penetrance. So I don't know how familiar you are looking at pedigrees or family trees, but the uppermost is the grand parental generation, so the most elder generation, and then you see everyone on a horizontal line of siblings, if they have a high bar, and people who are married are connected in the middle. So let's say this was colon cancer, you see, or breast cancer, and you see generation after generation affected. You see about half of the people have the cancer and half of them don't. The little dots in the middle are people who have not manifested any disease. So this woman who's had 3 affected children, she herself has not had cancer. So this person is a carrier, susceptible carrier, but may never develop cancer, or may have done something that, as an intervention, that changed her risk. Maybe she had her ovaries out when she was 35 without even knowing that it would make a difference in her breast cancer risk. And then the green box is the sporadic cancer. So we always want to confirm when there's hereditary cancer in a family that everybody who has a common cancer indeed is carrying the hereditary mutation, because about 5% of the time we see that they just happen to have a sporadic cancer. This is in particular with breast and colon, which are very common cancers in the U.S. And you wouldn't want to be telling that person's children that they're at risk for hereditary cancer if they're not. So here's a sort of a short list of common genetic testing for hereditary cancers. In breast and ovarian and colon and endometrial and prostate are certainly by far the most common. There's some endocrine tumors, kidney, stomach cancers that all have significant hereditary components. But cancers can cluster in a family and can make it look for all the world like there must be something genetic going on, but may in fact be a shared environmental risk. The most common of these would be lung cancer or Hodgkin's disease, which is probably due to a viral exposure, skin cancer, susceptibility, and obviously living in sun conditions. So we generally say 5 to 10% of both breast and ovarian cancer is due to a single gene, hereditary susceptibility. And then another 15 to 20% of breast cancer has this familial vulnerability. that you see more cancer than you'd expect by chance, that there are multiple generations, but it's not to the extent where it's dominant in terms of its pattern, and we can't find a single gene. This slide is from a nice review. If any of you are ambitious enough to sort of read kind of the 50 years of hereditary cancer research. And it kind of takes you through from 1969, which is that hypothesis of the 2 hits that was required for these cancer susceptibilities, to the seminal detection of a whole bunch of genes, all related to different kinds of hereditary cancers. You see that BRCA1 and BRCA2 are right in there in the mid-90s, and then some other familial cancer syndromes, both related to breast cancer as well as other cancers. And then in the more recent years, the last 5 years, been a lot of concentration on what's called GWAS, genome-wide studies looking for susceptibility to breast cancer, ovarian cancer, almost everything, and trying to see, what are the other genetic attributes. Of the known breast cancer genes that cause well-defined syndromes, there are less than a dozen, although we're finding a longer list that accounts for a very small percentage of certain families. Importantly, you see that BRCA1 and BRCA2 are very big actors in the hereditary cancer world, but by no means explain all of hereditary breast cancer. And this is a critical thing because I think that there continues to be a lot of confusion in people who are tested and BRCA negative, what the implication of that is for themselves and their cancer risk and for other members of their family. These smaller slivers of pie are a combination of a whole bunch of other known genes that range in their cancer risk from 30 to 40% lifetime to almost as high as that in BRCA. And then the biggest remaining piece is what's sort of undetectable in this combination of what we think to be these variations, these small hit. You have to have a be dealt a genetic load, and you need 10 or 15 or 50 of these little tiny variants in order to get the threshold of risk for what was traditionally called sporadic cancer. This is just for your reference. These are some cancer syndromes. Should you come across these names, these all have classic features. Cowden syndrome, Lee-Fraumeni, hereditary non-polyposis colon cancer, which is also called Lynch syndrome, Peutz-Jeghers. Either they have something you can see on the outside, clinical manifestations to their skin, benign growths or lesions that are very classic, and then a whole constellation of cancers that make them very unique. And all of those are available for single gene testing. Most of these can also have a breast cancer risk associated with them, or ovarian. And this slide is just to show you that in all of those genes on the prior slide, almost all of them are within this DNA damage and repair pathway. So we have a lot of redundancy. It is so critically important that our DNA, every time the cell divides and copies, that it's copied exactly the same, that it is perfect. Because if it's not perfect, then it causes change. And that's what a mutation is. So there's a tremendous amount of cellular effort placed into protecting the DNA being absolutely sterile in terms of its replication. The BRCA is within the double-stranded break, so repair of when the DNA is actually broken all the way through, and some of these other areas of injury all lead to other hereditary susceptibilities. So let's talk for a few minutes about the BRCA-related cancer risks. The lifetime risks, there's a big variation, and I know you guys had some statistics in your last course, but it's really a challenge. It's a challenge for clinicians. It's terribly challenging for patients. What is the number? Because if my cancer risk is 87%, I might make a different decision than if my cancer risk is 55% for breast cancer. And there really is no absolute number. All of those numbers are based on studies that have had different designs, that have different populations in them, and that some of them have built into it significant biases or errors. So you have to recognize it as a range. When I talk to patients, it's much more meaningful. I break it down into sort of 10-year risks because those are pretty static. The risk, and I'll show you in a minute some tables, that the risk, regardless of what the lifetime is, it depends on whether that lifetime is to age 70, if that lifetime is to age 80, and the amount of surety that they have in the populations that they're looking at that everybody had a mutation. But that aside, BRCA1 is mostly breast and ovarian cancer, female breast cancer and ovarian cancer. It's associated with very high risk for both. The ovarian cancer risk is anywhere from up to about 50%. The breast cancer risk is skewed to early age of onset and very typically highly aggressive triple-negative estrogen hormone non-responsive cancer and a high risk for second breast cancer. BRCA2 has breast and ovarian cancer. The breast cancer tends to be later. The breast cancer tends to mirror that of sporadic breast cancer with estrogen and progestin hormone-sensitive tumors that have other medical options available to them for management. Has a significant male breast cancer risk, which is 6 to 7%, which is close to what the sporadic breast cancer risk is in a low-risk woman. BRCA2 also has a substantial prostate cancer risk. The ovarian cancer risk is significantly lower in BRCA2. And in BRCA1, it's like 25 to 50% or 55%. In BRCA2, ranges from about 10 to 25% from the low to the high end. So here's a bigger pedigree, and this is sort of what we look at when we're analyzing family histories and to say, does this appear to be hereditary breast ovary syndrome? So we talk about close relatives or degree of relationship. So first degree are your parents, full siblings, and children. And second degree, you move out from there, grandparents, aunts, uncles, nieces, nephews, whatnot. So the closer you are, the closer the number of relatives are. Often it's asked about first-degree relatives. Do you have a mother, a sister, or a daughter with breast cancer, for example? Now, the fault in that is that what it loses is the paternal family history, because by virtue of having the first-degree relative being male, everybody else is a second degree or further. But males are often silent carriers for BRCA. So it's really very important to extensively look at the paternal family history, and there's been a lot of effort in trying to better allow, particularly from an insurance standpoint, to recognize that the degree of relatedness is different from the mother's and father's side. So this is a classic family. You see this early onset breast cancer in the first generation at 45, but she then went on and lived a long time after that. She had no children. Then there is an elderly patient with no cancer who has a daughter who had ovarian cancer, and then a son whose daughter in turn had an early onset breast cancer, and so on. And an important thing in BRCA, there's often sort of this coined term, sort of the Jewish genes for breast cancer. And it always bothers me because BRCA is a mutation that occurs in every corner of the world. The importance of kind of the ethnic distribution in the next few slides will show you that there are these recurrent mutations in populations that are more isolated. So there are almost no illnesses that are unique to any one group of people. And that overrepresentation either may be because the carriers had some kind of health advantage, that it's a very isolated community, a closed reproductive pool for social reasons, or there was some kind of population bottleneck where there was a significant reduction in the population size, and then it regrew out of a very small number of founders. So when we talk about the carrier frequency, we're really saying how often is this occurring in the population. And there definitely are differences in the carrier frequency from one ethnic group to another for BRCA, with one of the highest being in Eastern European Ashkenazi Jewish people, but that this frequency is mirrored by other populations, as you'll see in a minute. But all basically you do is you add up the number of people and then the number of people that have the mutation, and then that fraction becomes the carrier frequency. So in this slide, it's 2 out of 10, and the carrier frequency is 20%. With BRCA1 and BRCA2, in the Eastern European or Ashkenazi Jewish population, there are 3 common recurrent mutations, 2 on BRCA1 and 1 in BRCA2. But they're also found in a whole host of other communities. The prevalence altogether in the Ashkenazi Jewish population is about 1 in 40. So this is a slide showing what's called the diaspora. It's the migration of the Jewish people out from the primordial people in the Middle East out to Eastern Europe and founder populations from there. But here are significant founder mutations worldwide, and so if you just sort of peruse this, you can see that there are repeated mutations that are seen all across the globe, some of them with high frequency. And this slide is sort of confusing. There's some early data that basically was done by Myriad, who owns the patent on the BRCA genes, and they showed the prevalence of mutations in people who met the clinical criteria. And the only thing to point out here is that, if you are set based on meeting clinical criteria, the mutation rate is about the same in all ethnic groups. What about missed BRCA mutations? So early on, about a decade ago, there was an innovation to the genetic testing when there was the realization that sometimes the errors are not just single changes when we looked at that sentence, but in fact, they were large rearrangements. So if the gene is broken in 2 places and then flipped upside down, if you're in between, if you think of that sentence, the big red dog, if the break is between the word the and the word dog, nice and evenly, and then reversed, then all the letters, if read by sequencing, are in order, but it still interrupts the function of the gene. So there was the first addition to the genetic testing was in about 2001, and then continued linkage studies show that clearly people seem to have mutations in BRCA that we were not able to find. So in 2006, there was a big study of the BRCA3 or the mystery gene, and what they found was that they were able to find another 7% of people had additional rearrangements looking by another technology, and that 5% had mutations in a gene that I showed you in the list of genes that's a common mutation in Caucasians, in European Caucasians. So this additional testing is referred to as BART, or genomic rearrangement, BRCA rearrangement testing by Myriad. For a long time, since 2007 to now, it was a breakout or a separate test unless the individual met very, very strict criteria for hereditary breast ovary syndrome. There's a lot more data available now, and we know that it accounts for mutations, significant amount of mutations in minorities, and is very important in testing for those. Does not make up a lot of mutations in the Ashkenazi Jewish population, but it is clearly most appropriate for those who meet the classic criteria. But it's been very challenging in terms of getting that additional testing paid for and who should have it and who shouldn't. But there's a lot of movement now in terms of insurance companies covering this testing. Variants are something that's also very problematic in any genetic testing, and that's when we talk about those little missense, so that little substitution of one nucleotide for another. Does it mean anything or it doesn't? So if it's the first time that that's been seen in the individuals, the first time ever reported, it's very difficult to know whether it has any consequence. So when genetic testing first started, this was a huge problem. And then there's been a sort of worldwide effort to put data out there so that everybody could share how many times the mutation, this variation has been seen, if there have been any laboratory analyses to show that there's no function, does it track with cancers in the family, a lot of data to try to reclassify them. So if a person's genetic testing showed a variant of uncertain clinical significance, it may be that in 6 months or 10 years that could be reclassified. either to say this clearly is a BRCA mutation that increases susceptibility and should be used clinically, or means nothing and will no longer be reported. So this is an area where people also get into big difficulties, both clinicians and patients, in trying to understand, what does that really mean, because they have a piece of paper that shows some kind of genetic change. So this is a slightly busy slide showing the testing algorithm and kind of working through how much testing should be done, what does it mean, when do you say that negative is negative, and when do you give reassurance that that person is no longer at high risk. And it's maybe beyond sort of the scope for today's discussion, but just recognize that also for, let's say, somebody who has a high founder mutation, so Ashkenazi Jewish or French Canadian, if that first-tier testing is negative, but that person comes from a high-risk family and that is very compelling in terms of that family history, additional testing is very appropriate because there may be a non-founder mutation. In the Ashkenazi Jewish population, this occurs about 5% of the time. So who should have genetic testing? Here's sort of just a short list of the very clear indicators that meet every insurance criteria. What's been added that's new is that Medicare now is considering triple-negative breast cancer, so the estrogen, progestin negative, HER2 negative tumors in a younger patient to be a single indicator qualifying for BRCA testing. And so that's something that's very new in the last year. But otherwise, these sort of classic lists, particularly male breast cancer, bilateral breast cancer, early age of onset, breast and ovarian cancer. Ovarian cancer by itself has such a high frequency of mutations that in the Caucasian population it's about 10%, and in the Ashkenazi Jewish population it's 30%, even if you have no family history. That everybody should be offered testing, in my opinion, who's had ovarian, fallopian tube, or primary peritoneal cancer. So the walk-off is, half of BRCA mutations come from the father. So in families that look very quiet, that there is no family history, it's often male to male to male before it finally gets to a woman. Ovarian cancer, like I just said, very important indicator. That early onset breast cancer and bilateral breast cancer is more important than having a lot of family members with late onset breast cancer, which may very well be genetic and maybe, but likely to have a different cause than BRCA. So what are common misconceptions? That paternal history doesn't matter, that hereditary cancer, hereditary breast cancer equals BRCA, and then a whole reason, a whole list of things in terms of trying to understand if you're BRCA negative, what does that mean? On both sides, there was some literature that suggested that even if you were BRCA negative and came from a BRCA family, that you might still have a higher risk of breast cancer. This has been put to rest. Or that if you're BRCA negative, but there's lots of breast cancer and ovarian cancer and no mutation has been detected anywhere, that she's now still at population risk, which neither of which of those are true. Okay. So critical to what I do and critical to anybody receiving genetic testing is the process of genetic counseling. And genetic counseling is a subspecialty. It's a master's level profession that work in conjunction with geneticists like myself that really take this very complicated information and translate it to a way that patients can understand. The basic tenets are non-directive, so nobody should be told they have to have genetic testing. It's always a decision of the individual, which is very different than a lot of other aspects of medicine where you go to the doctor and they say you have to have this cholesterol test. A lot of assistance in terms of getting records, interpretation, and then management recommendations, and sort of an ongoing resource, as well as, importantly, information about concerns for discrimination, how to talk to family members, etc. So I want to just spend a couple of minutes talking about the next generation of genetic testing, because of all of you who are going out as advocates, you're going to hear a lot about, sort of what's new and what's out there. So one thing which has just come sort of fresh off the press and is now becoming clinically available since just the spring is combining a lot of these genes that I showed you in that pie and various slides, all onto one platform so that we can test for them all at once. So this is a break from the prior kind of dogma of saying we're going to choose one genetic test based on the construct of the family history. That's not to say that that's not important and not probably the best way to still approach it, but what we found is that the more you look, the more you find. So in individuals who may be the only person in their family with an early onset cancer, or there are lots of men, we're often unable to make an adequate interpretation of the family history. So there was a series of studies done by the Mary Claire King's group looking at cancers that just sort of in sequence. And they looked at 20 genes, for example, for ovarian cancer. Just to move you through this slide, what they found was 360 subjects. They were not selected for age or family history, and a quarter of them carried germline mutations. 20% almost was in BRCA1 or 2, but then another 6% had a whole variety of genes, of which very few we routinely clinically test for ovarian cancer. And that was the part that was really shocking to those of us in clinical hereditary cancer. A third had no family history, and over a third were over the age of 60. So they would have met no diagnostic bars that we've kind of had long-standing. And so now this is becoming clinically available, and there's one lab that's currently offering this multi-gene panel for breast cancer, colon cancer, ovarian cancer, and if you have multiple cancers. It varies. It is costly. The insurance coverage is just sort of being tested and assessed on a case-by-case basis. Is this to say that everybody should have this testing and not go stepwise? Absolutely not. But I think that ultimately, because the technology will allow for less and less expensive genetic testing, we're going to have the opportunity to look more broadly at first. The potential downside is when we were talking about those uninterpretable variants. If you increase the number of tests by 20, the likelihood that you're going to find something that you have never seen before and don't know what to do with goes up by 20 as well. And so that's where a lot of caution needs to be garnered there, as well as a lot of these genes we actually don't have very good data to say, well, okay, you have a mutation here, but we have no idea what to tell your family in terms of what their risks for cancer are. So this is a table that I was talking to you about, and this is Joy Morris, and some of you may know Joy. She's a very active member of FORCE and a local coordinator. And she and I wrote a book together really for patients to try to understand BRCA. So this table breaks down by decade. This would be the patient who has no personal history of cancer, their current age, and then going out for what's the next 10 years. And it's based on an accumulation of 10 different studies. And so what you see is that the younger you are, the longer you go out to age 70 for BRCA1 and BRCA2, these are the lifetime risks. But if you're 60 and you go out, what's the risk for cancer in the next 10 years from the age of 60 to 70, then it's a fraction of that. So at the age of 60 or 65, you don't still have the, whatever the number is, 60% or 80% lifetime risk for cancer. The reason why some of these numbers, the lifetime risks, are significantly lower is that this is to age 70. Breast cancer in everyone is a disease of aging, and that studies that go out to age 80, that number just continues to climb. And the same for ovarian cancer. You'll see here the same thing in terms of the lifetime risks, 40% by age 70, 16% in BRCA2. Male breast cancer in BRCA2, 7%, about 7% risk. It's always repeated that there is a breast cancer risk in BRCA1, but there's really no cancer risk number associated with it. The light pink is the population risk, and the purple is the BRCA 20% risk for prostate cancer. It's beyond the scope of today's talk to talk about managing the risk. What do we do? But suffice it to say, we have proven interventions that there is benefit both in terms of longevity and morbidity or reduction in burden by enhanced surveillance with early detection, what we call chemopreventive with hormone manipulation and prophylactic surgery, both breast and ovarian cancer. BRCA testing has become very important as point of care. So if a woman is diagnosed with breast cancer, determining whether or not she has a BRCA mutation often dictates what kind of treatment she has, because the risk for contralateral breast cancer in BRCA carriers is 30% at 10 years if you don't do something. A lot of women will then elect to have bilateral mastectomy, but by no means does everybody decide to have that as their treatment of choice. Taking tamoxifen and having your ovaries removed, particularly premenopausal, the earlier the more significant, significantly reduces the risk of contralateral or a second primary breast cancer. So in close, in terms of the red flags for hereditary cancer are early age, ovarian cancer, male breast cancer, multiple primaries. It is certainly Eastern European Jewish ancestry with breast or ovarian cancer, and obviously if there's a BRCA mutation already known in the family. So we've talked about this already for a little bit, but I just wanted to again sort of have you reinforce this idea of the polygenic or the genetics of common cancers. We haven't found a BRCA3. We found a lot of other genes that in certain families are the modest to higher risk susceptibilities, but they really don't, they're not the big actors. They don't account for enough globally, maybe 1 to 3% of familial breast cancer. So there's been a lot of effort looking at what's called low penetrant, little genetic effects that you'd need to have in aggregate in order to have this vulnerability. And this is called the polygenic model of cancer. So if you're stacked and you have a whole bunch of these high-risk little alleles, you're more likely to get breast cancer. If you're stacked with lots of low-risk alleles, then you're very unlikely to get breast cancer. So that's what the construct is, and that's what all these genome studies have been trying to look at. Why do they work? Because these variations tend to be two forms. You have the A form or the B form. So that you can look at groups of people and say, if you have A, are you more likely to have disease than if you're in B? And then they are in something called a haplotype, so that if you have A in one location, it can tell you that for the whole next section of the genome that things tend to look exactly the same, that they're all linked together. And these occur about 1 every 300 to every 1,000 base pairs. We have this variation. And so here you see that if you looked at this combination, you would expect that for these 6 different polymorphisms or variations, that if you multiply them, that there should be 64 different combinations that could be possible. But in fact, when we sequence lots of people, there are only 4, because each one dictates pretty specifically the whole region. If you have this, then this is your pattern. If you have this, then this is your pattern. So what are the barriers that we see to genetic testing? Genetic counseling is critical. Most of the errors in information or misinformation is related to people who really don't understand the genetics of hereditary cancer or genetics at all, and that they are then giving genetic testing and interpretation of the genetic testing. Almost all insurers pay for genetic testing. There are rare exceptions where there's an exclusion. All of the national health care, Medicare, Medi-Cal now also pays for BRCA testing. But there's still a lot of cultural perception about causation, about stigmatization in the family. And despite the fact that there's very good federal legislation which has been enacted for over 1.5 years, there's still a lot of fear about genetic discrimination. So GINA was the Genetic Information Non-Discrimination Act. It was passed at the end of 2009. It rolled out in 2010, and we've been collecting data since then. And it's really broad, sweeping protection for both employment and healthcare protection from susceptibility discrimination. So if you have had breast cancer or ovarian cancer, the way the underwriting works in this country is that you can then be still categorized as high risk, and you can have exclusions and you can have pre-existing. But if you are a healthy individual who is found to be at risk, then you cannot be discriminated against. And it's mandated to pay for screening, preventive surgeries. All of this is considered customary care. And the flip side of that, as I always tell providers, is that there's now also precedent in terms of duty to warn. That it's not to say that practitioners, if talking to a patient and the patient says, well, I don't talk to my sister and I don't care if she knows that I have a BRCA mutation, that physician is not obligated to go out and find that woman and tell her that there's a BRCA mutation in the family, and it's by no means a reportable disorder. But the duty of warn goes to the point where the physician has to explain who's at risk and provide the opportunity to be as helpful as they can, whether that means writing a letter, trying to explain that, whatever needs to be done in order to help facilitate communication to family members at risk. So with that, I'm going to close and open the forum to any questions, and I am very happy for this opportunity to be speaking with you tonight.
So, ladies and gentlemen, do we have any questions for Dr. Gordon? I think she's covered some fantastic, informative topics for us, and I'm wondering if anybody out there has anything on their mind they'd like to ask. I think nothing's come in yet, Dr. Gordon, so I'm just looking. Okay, here we go. Question. Let's see. So this is, I was shocked to see laryngeal cancer listed as one of the cancers associated with BRCA2. My mother died from this type of cancer. Can you give us more information, including the statistics associated with this type of cancer?
So that is a very good question. It's definitely been one of the cancers that's sort of one of the outliers. So esophageal, laryngeal, in the early studies of hereditary families, those cancers were overrepresented. Since then, there has been really no good data in terms of the BRCA association or not. One of the cancers that have been looked at more carefully has been colon cancer, which also was initially reported as an elevated cancer in BRCA, and in more careful analyses, Mark Robson and Tim Revick, they felt that it was, common cancers occurring commonly. So I don't have a great answer in terms of what's the absolute risk, or is it really an association, or was it a bystander that just happened to be in those early families? But it's certainly something that catches my attention and the cancer genetic counselor's attention if we see upper GI or laryngeal cancer in a constellation that otherwise looks like BRCA. It's not part of any testing criterion. So if you went to an insurance company and said, my mother had laryngeal cancer, I'm worried about BRCA, they would say, we don't think that that's a cancer associated with BRCA.
Interesting. What about colon cancer? We have somebody who asked about the fact that she read that people with a BRCA2 mutation are at higher risk for colon cancer. Is this true, and if so, are there new recommendations for colorectal screening?
So we just, that was sort of what I was just referencing, that there's been this evolution away from recommendations towards early onset screening for colon cancer, or really giving an attributable risk related to BRCA for colon cancer. That said, what I recommend clinically and can usually get justified is that, first of all, if there's any colon cancer in the family, you still use that as the bar, and you say you start screening 10 years earlier whenever that colon cancer was. In families that there is not a history of colon cancer, I like to try to get the baseline colonoscopy at 45 and then repeat it about every 5 years if there are no polyps or abnormalities detected.
Great, thank you. Okay, so now they're rolling in here. Let's see. Those of us who, with a BRCA mutation, start with one damaged gene. If we get cancer, does that mean that we don't have any good tumor-suppressing genes left to help us fight off other cancers?
So this is the part that's so hard to kind of get your mind around. when we talk about that second hit, it is only in the cell that ultimately, if it progresses to cancer, that's the cell. But remember when I showed, and I can see if I can go back, that slide that, I hope nobody gets nauseous with this major reversal here, that looked at, what are all the mechanisms that we have to protect our DNA. So we have a lot of redundancy. All of the genes, all of this series of genes here are all working to protect the DNA from further injury. But if in that one cell there's the second and third and fourth hit, then that may become the cell that can become cancerous. All the other cells in your body are fine and have to have what's thought to be, the 1 in a million hit that they get that radiation or that second damage. It's just that it's more likely that you get those additional injuries, which is why the risk for a second new cancer is so much higher in BRCA than it is in sporadic cancer.
Okay, great. Let's see. If there seems to be a strong family history of breast cancer, yet all tested members are BRCA negative, is there likely to be an elevated risk of ovarian cancer?
So that's a really good question. And there have been studies that have looked at that. And for the most part, that one of the powerful things, and I thank whoever asked that question, it's an important thing that I usually try to bring up, is that if all BRCA testing is comprehensively negative, then we provide a lot of reassurance that there is no elevated risk for ovarian cancer. The breast cancer risk may still be elevated and that we haven't identified that, but the only known association between breast and ovarian cancer at the current time is been BRCA. There's one exception to that, there's now a gene called PALB2, P-A-L-B2, which is a partner to BRCA, and so it can look, the constellation of cancers can look exactly like BRCA2, but it accounts for a very small proportion of hereditary families. And we'll see, now when we start to do this bigger panel testing and we start looking at all these genes we never looked at before, whether there's a change in that dogma or that philosophy. But right now, if you're BRCA negative and have been comprehensively tested, we say no increased risk in ovarian cancer.
Okay. We have some rather complex questions here that I think may have to be answered offline. But let's see, we'll hit a couple more. Besides the Ambry Next Gen testing, are there any other new types of emerging sequencing techniques applicable to BRCA on the horizon?
So in terms of what's the next generation in BRCA, at this point we're pretty confident that with the MLPA that we are capturing just about all mutations. I think that if there's going to be anything, it's going to be a recognition of another gene or an influence that silences the BRCA, that the BRCA itself is intact, but that what turns it on or off, that there's some abnormality there. But I'm not aware of any research, preliminary or otherwise, that has identified anything to that regard, but we found it in other hereditary cancers.
Okay.
Otherwise, it's sort of now really looking towards this genome-wide, trying to find really looking towards the common low penetrance as being the next answer.
Great. Let's see, regarding the statement on GWA, it's stated that CLL is one. If a mother, aunt, cousin, and grandmother all had CLL or leukemia and were Ashkenazi Jews, how would you view that?
I'm not entirely sure of the question, so...
yeah, okay. Regarding your statement on GWA, it stated CLL as one.
The studies in terms of the genome-wide studies for vulnerability to leukemia, if that's the question, there certainly have been studies looking at what is there vulnerability to leukemia. So we have not generally thought of leukemias as hereditary with a dominant, passed from one generation to the next. But there have been some studies looking at other vulnerabilities in bone marrow development that might make people more vulnerable or less. There's nothing in terms of a genetic test at this point that would identify risk for CLL.
Okay. Let's see. There was a question about genetic discrimination and insurance, and I can tell I'm not going to burden you, Dr. Gordon, with that. We have a significant amount of information on the GINA law on our website, and I think we can provide some additional information for that in our next email to the group. So we'll make this, I think, the last one for this evening. If I know that I have a BRCA mutation but have breast and multiple other cancers on both sides of my family, is there any benefit to additional genetic testing? So I guess knowing that there's a BRCA mutation on one side but there's a cancer history on the other, is there benefit to testing the other side of the family?
All right. So for that person as an individual, generally, the BRCA risk trumps everything else. So from a intervention and surveillance standpoint, that's probably all that person needs. But from a clarification, if there were maybe other cancers, so let's say like colon cancer, that was a vulnerability coming from the other side, that would be something that you'd want to identify. And then for other family members, so for identifying risk, you could be lucky enough to carry a BRCA mutation and some other susceptibility that you got from the other side. And so for passing that on to your children, identifying that, clarifying that risk so that you know, in fact, what they may be at risk for would also be helpful.
So I'm just curious for my own purposes. I know a situation where an individual carried both the BRCA1 and the BRCA2 mutations.
Right.
And that is obviously important information for her, but also potentially for her children. So if one suspects a BRCA mutation on the other side of the family, but maybe perhaps has not undergone full sequencing, would it be advisable for an individual to do that?
Yeah, so that's what I would recommend. So there are two things. One is that, let's say, some of the differences, even with just BRCA, that could be coming from both sides. You can carry a BRCA1 and a BRCA2 mutation. It does not make you at higher risk for cancer carrying those two. It just makes you at risk for the spectrum of both. So the BRCA2 that has male breast cancer and pancreatic cancer obviously has bigger implications for men, melanoma, some of the other GI cancers, which are these loosely associated with BRCA2, so that's important to know. And then the BRCA1, which has that much earlier onset and more aggressive, so timing of when you might do interventions would make that difference. So you kind of have that gamut. Because of how common in Eastern European Jewish people these mutations are, even if there's a known mutation on one side, I always test for the 3 common founder mutations. And if there's a compelling history on the side that we don't know about, I do full sequencing.
Interesting. Okay, one final question, because I think it's a good one. Do you think screening guidelines for BRCA should be expanded, especially in light of fathers who can be silent carriers? For example, in a family, a father has had non-Hodgkin's lymphoma, his sister had breast cancer, and dad had prostate cancer, but that family history was not enough for them to receive BRCA testing under the existing guidelines. Yeah. How would you view that? Do you think that maybe?
I think it will continue to liberalize. I think that we already see momentum towards relaxation of those strict guidelines. I think as the more genetic testing that happens and the more mutations that are found, will break open a lot of that, especially as the cost comes down. In that paper where I showed you the kind of seminal paper showing that there are these mutations in people that you'd never have suspected, they show a few case examples where the mutation that's found would never have been on anybody's radar. So in that regard, I think that will really put a lot of pressure to say, you have to justify the ability to do broad genetic testing. What I do in my clinic and what my counselors do is that we write strong letters, and what we find when insurance companies have denied, and then we take it up to medical review, and we give an articulate description, and with prostate cancer we can give some data in terms of frequency of mutations, they generally will approve it. But the person checking the boxes, they deny. The other thing is that there's a big movement towards perhaps universal screening for BRCA among high-risk populations like French Canadians and Ashkenazi Jewish individuals. There's been several studies showing that it's, people can understand even if they don't have a family history and that this is information that they want. So we may go towards that there may be no need for family history at all and ultimately be able to screen for those.
Great. Okay. There was a question about whether or not Myriad does the comprehensive panel of genetic testing, and I think I can answer this in that Myriad does both the founder's mutations, the full comprehensive panel, and the BART rearrangement. Is that correct?
That is correct.
Okay. And they are the only company currently doing those 3 types of genetic testing.
Right. They own the patent. It's patent protected in the U.S. and in Canada, although in Canada there still is centralized national testing that you can get done. So until 2014 or until the patent is overturned and it's been embroiled in a lot of legal battles, that's the only place you can get it. But it depends that they won't go on and do any additional testing. It all depends on the provider and what they order and how they order it. So there's a lot of responsibility on the clinician ordering that test to actually know what they're asking for.
Mm-hmm. And that goes back to, I think, why FORCE very much advocates for genetic counseling, so that the appropriate genetic tests are done and are recommended by somebody who is trained in knowing exactly what tests need to be done and what to look for. So with that, I think we're going to conclude the webinar this evening. There were some additional questions that we will be able to answer very easily offline on the website, more generic questions, and then there were a few with very specific personal stories that we try to keep our questions for the webinars a little more general, but we can try to address the more personal questions offline. So I'd like to thank you, Dr. Gordon, for a wonderful, wonderful presentation and for being such a big supporter of FORCE and our community. And I want to thank everybody for joining us this evening. So that concludes our webinar on genetics. Thank you very much.
My pleasure.
Have a good evening.